phenomics technique is highly promising because it reflects the end products of gene environment interactions. So the idea of systems-wide studies should have both phenomics data for individuals collected at multiple time points, along with exposure variables measured in detail, especially diet and what's the longitudinal follow-up. I think those results can have relevance for clinical and public health translations in terms of prediction and potential personalized prevention and nutrition. Thank you very much for your attention.
Thank you for this presentation, and for the sake of the time, we'll move with the next speaker.
Thank you.
Okay, the last speaker, last but not least.
I have a question.
There are no questions. There are no questions. In the interest of time, you're welcome to come speak to the speaker, but there are no questions, sorry. So I'd like to introduce Professor Valerio Orlando, and he has a long 20-year history in the genetic field. He's had a lot of contributions, including pioneering using the CHIP assay in vivo, and he was previously in Rome as a senior scientist at the Teleton Foundation. And then in January, he joined KAUST to direct the Environmental Epigenetics Research Program. So he is going to be speaking about epigenetic control of cell identity and plasticity. And if his slides appear, I think we'll have his talk. Yeah, they switched the computers. There it goes. Okay, that looks good.
First of all, I'd like to thank the organizers for inviting me here and contribute for this really important meeting. Although I didn't have to travel that long, so I'm not jet lagged. Right. So I think I don't need to introduce more about epigenetics or the epigenome. Just to remind the fact that the genome has a problem here, has to somehow interact with the environment, maybe also vice versa. And in order to interact with the environment, that's what the epigenome is good for. This is where it comes into play. This whole mediating the biological experience allows the genome basically to learn, memorize its experience, adapt, and then it takes it to stress, metabolism, social behavior, and diseases in various contexts. So epigenome actually is composed with various levels of organization of the genome in the nucleus here. And I can skip this. What is important here is that shape is information. So all these levels of organization in the genome, starting from the very basic chemical modification at the histone level up to the higher order organization, somehow reflects the ability of the genome to acquire different shapes. But shape is information per se. That's the kind of game that the epigenome likes to play in order to record and reproduce biological experience and phenotypic variation at the end of the day. So we're all happy with the notion that from one genome, thanks to the epigenome, we basically selectively activate or repress genes and give rise to different, the various 250 cell types that are composed our body here. So to each of these configurations corresponds also a specific transcription program and cell identity. Now the issue here is how plastic is actually this configuration, how stable is the configuration, and what are the mechanisms underlying the ability of cells and epigenome basically to adapt from time to time to environment. So this is exactly the question that has been inspiring our latest work, basically understanding the mechanism underlying epigenome regulation in post-mitotic cells. The issue here is that most of the epigenetic regulators that have been identified along the years have to do, been identified in the context of development. But very little is known, and probably the work of Paolo is one of the unique examples that very little is known in how the epigenome actually it works in post-mitotic cells. So the model system we've been using to address this question is the polygon group proteins. So this is a long history behind it, but briefly, this comes from the cloning and identification of genes involved in the fly development. And in particular, in proteins that control the body plan and the expression, correct spatial expression of homeotic genes in the body of the fly. So this will be a normal expression or state of homeotic genes controlling the posterior part of the fly. In polygon mutants, you see de-repression throughout the gene body, and so messing up the developmental profile. Now, polygon proteins that actually give rise to multi-protein complexes. I don't have time to go into the complexity of this, but probably the most best characterized element here that mediates the function, the genetic function of polygon proteins, is the methylation of histone H3 and the residue lysine 27 here. This is a cell cycle regulated event. There is a component here called SUSI12 and the histone methyltransferase EZH1 or EZH2 that interact with histones and mediate this silencing function. Now, as I said, there's very little known about any of the genetic regulators in somatic cells, and in particular polygon proteins in mammalian cells in particular, and post-mitotic tissues is completely unexplored. So we decided to start this study, and as a model system we use mammalian skeletal muscle cell differentiation. So this is briefly depicted here. You go from satellite cells, proliferating myoblasts, ending up with myofibers. Very nicely, you can work this in vitro. You can follow several markers that are specific of the various stages of differentiation. And also follow the expression state of transcription factors that are involved in this process. Now, if we follow the components of polygon complex here, in particular these two potential histone methyltransferases, we see that in nuclear in particular, we forget about cytoplasm for the moment for a second, we see that there are these two isomers, EZH2, which is the classic histone methyltransferase. But this seems to leave the stage after a certain point. And this seems to be substituted by this other protein, EZH1, virtually identical to EZH2. Now this is identical to EZH2, also in its set domain, but somehow it's a very, very weak histone methyltransferase, and its function is very much unknown. A couple of papers from my lab and also from Vittorio Sartorelli at the NIH led to the following, let's say, intriguing point. EZH2 would be associated during muscle differentiation to repressed domains, whereas EZH1, while it's substituting EZH2 in the cell, it will go this way, basically to bind to active domains. We came up with different interpretations about these puzzling results, so namely a full repressor, polygon repressor sitting on active genes and not on inactive genes. So the question is, whether this polygon protein is, or EZH1 is an activator or repressor somehow. So we got curious about the complexity of EZH1 gene itself, and our attention was caught by this second transcript here in the region. The second transcript would then call for a shorter isoform of EZH1 as compared to the longer one. So we checked whether this would be coding for anything, and we were lucky enough actually to find good antibodies for this. Now both would contain set domain. Okay, they would differ for the present catalytic and set domain itself. But so the long one would have a very functional set domain, but the short one basically would not have the set domain here. So we got intrigued by this, and so we can skip this. Now, if we follow the expression of this short and long, thanks to the antibodies we have available that can discriminate the two isoforms, we see that both long and short increase during differentiation here. So in the myoblasts, myotubes at various stages also. So the two isoforms somehow follow each other. Now they are nicely co-expressed also in other tissues that we have written, the brain, liver, spleen, and others. and they is opposite to EZH2, which normally EZH2 is expressed in proliferating cells and not in post-mitotic cells. So, the other important thing is that EZH1 long isoform seems to be nuclear, whereas EZH1 is also cytoplasmic. So we find EZH1 in both compartments, both nuclear, but also cytoplasmic, in contrast to the long isoform. Now, then we challenge whether, we ask a question whether EZH1 short will be part of the PRC2 complex or at all. And so these are co-IP experiments show that clearly both isoforms actually have the ability to be part of the core complex of this repressive complex, PRC2. So, so we postulate the idea that then the presence of a repressive complex on active genes would in fact allow active genes to adapt to stress response and perhaps repress genes at convenience. Okay. So we challenged the system with oxidative stress here, which mimics basically atrophic conditions, or hypertrophic conditions, which goes in the opposite direction. So with atrophic conditions, normally what you see is a downregulation of most genes. With hypertrophy, we see upregulation of genes. So to cut a long story short, we see that there is a stimulation by atrophic stress of EZH1 isoform levels here. You see that myosin is repressed here, whereas it's, in the case of heat shock response, it's upregulated here. But you see that EZH2 is gone basically here, and EZH1 is either up or in this here, inside is here. Okay. So then if we follow H3K27 levels, so the target sequence, the target of PRC2 complex here, we see that correspondingly in atrophic conditions, we see the levels of H3K27 trimethylation being upregulated, and in the opposite side, the hypertrophic going down. This is basically the data. We don't have time to go too much into the details, but they say this condition here provokes this increase here. So this experiment shows that EZH1 is actually responsible for this increase. So if we pull down the complex EZH1 here and challenge this in an in vitro assay, we see that this is actually becomes active, whereas EZH2 is not active. So this is, we identify a physiological condition that somehow makes the complex active and not inactive as normally it is. So we follow then three groups of genes, asking the question what is the association of this complex with different sets of genes. In particular, those genes that during differentiation would stay off, those genes that would stay on, or those genes that from off state would go on an on state. And here, what we see is that the association of ACH1 follows exactly this dynamics here. So those genes that are on, they show basically ACH1, but those that goes with on genes, off-on genes also they show the ACH1 here. Okay. So this is mirrored by the dynamics also of H3K27 trimethylation during differentiation. So those genes that change their state, they also acquire H3K27 methylation, whereas those who don't change their state, they basically remain stable. And this is EZH1 dependent. Now, those genes that go from off to on state in response to atrophic conditions, basically they are repressed by long EZH1 isoform in mitochondria and not by the short isoform here. So this was a really intriguing result because it was telling that the two isoforms are there, but why is the long isoform the one really responsible? Who is the only one containing the histone transferase activity? So we suspected an interplay between the two isoforms. So in particular, we checked whether the short or long isoforms they will show any dynamics on chromatin. And these are experiments that show by chip basically that upon stress, we see the short isoform that is normally in normal conditions more prominent on chromatin. to leave the chromatin here and be replaced basically by the long isoform. Now we follow also the destiny of the short isoforms, and we see that this becomes ubiquitinated upon a stress response here, and we eventually transported in the cytoplasm and degraded. So we check then in vivo, so we use basically a mouse model for atrophic conditions here. Basically, we enable we impede the mouse to move the leg for a certain number of days or weeks, and then we let it recover. The take-home message here is that if we follow ACH1 long and short isoform regulation in this context, we see that at the time when the muscle is stressed in atrophic conditions, we see an increase in long isoform and a decrease in short isoform, increase in H3K27. But if we let the mouse recover from this condition here, we see that everything goes to normal. The two isoforms really shuffle each other. So this is the working model here. Basically, we see that in somatic cells, an inactive ECH1 isoform seems to sit on the chromatin on active genes. But upon stress here, what we see is that there is a recruitment of the active histone-methyltransferase, and correspondingly, the short isoform, which would, in this configuration, would inhibit the binding by competition on the core complex here, will leave the stage and eventually be degraded. So this is rather new and totally unexpected new mechanism underlying polycomb function or epigenetic regulator by an interplay between two different isoforms that alternatively are present in different physiological conditions in a reversible manner. So at the end of the day, whatever, and I'm closing here with 3 slides, whatever the epigenome is good for, in case of repression or activation, polymerase remains the main target. So the adaptation to stress, or the adaptation to any metabolic function or whatever, eventually culminates with the regulation of Pol2 regulation itself. So we and others have shown in the past that polycomb itself is actually interacting with the RNA polymerase 2 and so on. But there is another interplay pretty important that connects polymerase regulation, and this is non-coding RNA and also RNAi. So we published a couple of years ago the finding that RNA interference is actually acting also in the nucleus. It's actually in the nucleus by directly contacting Pol2, and this is particularly relevant in conditions where cells are stressed. Namely, RNAi is important to modulate, negative modulate Pol2 to counteract stress response here. Now the scenario becomes more complicated if we then look at the mammalian cells, and again here we find a short form of dicer that would be specific for nuclear compartment. Is in a similar way to what I will show for polycomb. This dicer is associated with Pol2 and is associated with chromatin in particular condition. Now, to cut the story short, we have evidence that dicer would basically interact with the non-coding RNA and somehow be implicated in modulating the non-coding RNA-mediated enhancer promoter response in mammalian genes. So, just this to give you an example, there are 2 issues here to study now the ability of epigenome to respond to environment. One level is the global level, so how the shape of the genome is actually organized in response to stimuli, and this is completely unknown. And also the proteome is how this changes in various physiological conditions in post-mitotic cells. That's what is actually inspiring our future direction, so basically to explore how the epigenome dynamics in adaptive response as a general theme, so looking at transcriptome, proteome in different physiological conditions, looking higher at the structures, and also looking at the contribution of repetitive elements. I didn't have time to go into this, to genome shaping in general. What we suspect is that the cohort of new molecules will be present depending on the physiological conditions that would mediate the ability of the epigenome to respond to environment. I'm closing here, featuring now this big transition from Rome, Italy to KAUST for the creation and launch of the new KAUST Developmental Epigenetics Program that I do hope will serve the kingdom and the students and all collaborations with any other, in particular our neighbors cow colleagues here in Jeddah. Thank you very much for your attention.
Okay, thank you, Valerie, for a great talk. In the interest of time, I think there will not be questions. We've run over a bit. I apologize that there couldn't be questions for the last two speakers, but they'll be available tomorrow and perhaps a little time today if people have questions. So thank you very much.
Well, thank you very much, and I would like to thank the chairpersons of the last session and everyone who has stayed with us till the end. Thanks a lot, and we'll see you tomorrow. Tomorrow the program will be in the smaller conference halls. The one on the periphery, and there's a poster session